children. Last class we have discussed with the mutation breeding. I have started with today polyploidy breeding. Majority of flowering plants are diploid, that they are having diploid number of chromosome in its somatic cell. The plant which possesses more than 2 sets of chromosomes are called polyploids. What is called polyploids? The plants which possess more than 2 sets of chromosomes are called polyploids. Polyploid is a major force in the evolution of both wild and cultivated plants. Polyploid often exhibit increased hybrid vigor, increased heterozygosity, increased tolerance to both biotic and abiotic stress, buffering of deleterious mutations. So these are the advantages of polyploid breedings. 2-mark question. I mentioned the significance or advantages of polyploid breeding. Polyploid breeds exhibit increased hybrid vigor, increased heterozygosity, increase tolerance to both biotic and abiotic stresses, buffering of deleterious mutation. In addition, polyploid often results in reduced fertility due to meiotic error, allowing the production of seedless varieties. Okay, so but polyploids has a reduced fertility due to meiotic error. Because of that, it is producing seedless varieties. When chromosome number is doubled by itself in the same plant, it is called autopolyploid. Example, triploid condition in sugar beets, apples and pear has resulted in the increase in vigor and fruit size, large root size, large leaves, flower, more seeds and sugar content in them. So when chromosome number is doubled by itself in the same plant, and this condition is called the autopolyploid, and example is given. Triploid condition in sugarcane, apple, pear, etc. And it also resulted in seedless tomato, apple, watermelon and orange. Polyploidy can be induced by the use of colchicine to double the number of chromosomes. Then this is called what? Allopolyploids. Allopolyploids are produced by multiplication of chromosome sets that are initially derived from two different species. Example, triticale, Triticum purum, with Secale cereal, and Rhabdobrassica, which are produced by the combination of Brassica oleracea and Rabanus sativus. This is about what? Polyploid breeding. And next one is what? Green revolution. What is green revolution? The term green revolution was coined by William S. Gott in 1968. It is defined as the cumulative result of a series of research, development, innovation and technology transfer initiatives. Agriculture production, especially wheat and rice, manifold worldwide, particularly in the developing countries between 1940s and the late 1960s. So green revolution is a term coined by William S. Gott, 1-mark question, and its definition will be cumulative result of series of research, development, innovation and technology transfer initiatives in agricultural production such as wheat and rice is called what? Green revolution. The green revolution or third agricultural revolution is the intensive plan of 1960s to increase crop yield in developing countries by introducing the high yielding resistant varieties, increased irrigation facilities, fertilizer application and better agricultural management. So these are the various techniques they are used for increase the crop yielding, and it is called grain revolution. It has taken place during the year 1960s. In 1963, semi-dwarf wheat of Mexico was introduced from which India got 5 prolonged strategies for breeding a wide range of high varieties like Sonara 64, Sonalica, Kalyana Sona, possessing a broad spectrum of resistance to major biotic and abiotic conditions. Okay, in 1963, a semi-dwarf wheat of Mexico, that is a wheat which is introduced from the Mexico, it is a semi-dwarf from which India got 5 prolonged strategies for breeding a wide range of high varieties like Sonora 64, Sonalica, Kalyana Sona, which are possessing a broad spectrum of resistance to both biotic and abiotic condition. Same as wheat, M.S. Swaminathan produced the first semi-dwarf fertilizer responsive hybrid variety of rice TN1, that is Taiseng Native 1, in 1956 from Taiwan. Then their derivatives were introduced in 1966. So like wheat, MS Swaminathan produced a semi-dwarf varieties of rice, that is TN1 from Taiwan. From this they derived some other types of wheat, rice also. Later better yielding semi-dwarf varieties of rice Jaya and Ratna developed in India. So then this is our about green revolution. Norin 10. The cultivars found that Norin 10 dwarfing gene have a high photosynthetic rate per unit leaf area and increase respiratory activity. Then Konjiro Inasuka selected the semi-dwarf wheat variety that became Norin 10. He would have never thought that the semi-dwarf genes would not only revolutionize the world of wheat but also helped to save more than 1 billion lives from hunger and starvation. See that. Then plant breeding for developing resistant to diseases. Some crop varieties breed by hybridization and selection for disease resistant to fungi, bacteria and viral diseases are released. See crop wheat variety Kimgiri, which are resistant to leaf and stripe rust, gill burnt, and Brassica, it is a variety Busa swarnim, Kararai, wheat rust. It is resistant to disease white rust. Cauliflower is a variety Pusa subra, Pusa snowball K1. It is resistant to black rot and curl blight black rot. Then cowpea Pusa komal bacterial blight, chili Pusa sadabahar, chili mosaic virus, tobacco mosaic virus and leaf curl virus. Okay, and this box things all please you go through by yourself children. Definitely you will get some question from this box. And next one is resistant to yellow mosaic virus in bindi. Abelmoschus esculentus was transferred from a wild species and resulted in a new variety of Abelmoschus esculens called Barberni grantii. Plant breeding for developing resistance to insect pest. Insect resistance in host crop plants may be due to morphological, biochemical, physiological characteristic. Hairy leaves in several plants are associated with resistance to insect pest. Example, resistant to jazz seeds in cotton and a cereal leaf beetle in wheat. In wheat, solid stems lead to non-preference by the stems sawfly, and smooth leaves and nectarless cotton varieties do not attack bollworms. High aspartic acid, low nitrogen and sugar cotton in maize leads to resistant to maize stem borer. So same thing, disease resistant insect pest resistant varieties we will have in the table. Brassica, rapeseed mustard, a variety is called Pusa Kaurav and it is resistant to aphids. Next, flat bean Pusa sem 2, Pusa sem 3. It is resistant to the insect jazid, aphids and fruit borer. And next one is okra bindi. Its variety is Pusa savani and Pusa A4. Resistant to soot and brood borer. Modern plant breeding. In the milestone of plant breeding methods, genetic engineering, plant tissue culture, protoplasmic fusion or somatic hybridization, molecular marking and DNA fingerprinting are some of the modern plant breeding tools used to improve the crop varieties. We have already discussed about the various technique and application of the above mentioned concept in Unit 8 itself. new plant engineering technique, new breeding technique. New breeding technique or a collection of methods that could increase and accelerate the development of new traits in plant breeding. This technique often involve genome editing to modify DNA at a specific location within the plants to produce new traits in crop plants. The various methods of achieving these changes in traits include the following. First one is cutting. This is a 2-mark question, children. What are the modern new plant engineering technique? Discuss it. You will get as a 5-mark question. First one, cutting and modifying the genome during the repair process by tools like CRISPR or CASE. First what we will do during this technique, cutting and modifying the genome. Okay, and this is called what? This is done by using the tools like CRISPR or CASE. Genome editing to introduce changes in few base pairs using a technique called oligonucleotide directed mutagenesis. After cutting, we can editing the introduced changes in new base pairs using a technique called oligonucleotide directed mutagenesis, and transforming a gene from an identical or closely related species. Okay, after editing, we have to transfer the gene into an identical or closely related species. This is called what? Cisgenesis. And organizing processes that after gene activity without altering the DNA itself, this is called what? Epigenetic method. Organism process that alter gene. So after introducing this altered gene into the organism process that alter gene activity without altering the DNA itself, and this process is called what? Epigenetic methods. Okay. Okay, children, with this we have completed this lesson. So we will start the next lesson, that is 10th lesson of botany, in my next class, children. Okay, so we will go little fast. We have many lessons still there, so I have planned to go little bit fast. Please cope up, children, and complete the homework given then and there. And why girls are not at all submitting your test paper, children? Submit it soon. Bye. I will get you in my next class, and today's portion will be a homework for you, and complete it. Bye.